So in 14.24, we are going to, well, I'll read the problem. Shown below are several amino acid substitutions in the alpha and beta chains of human hemoglobin. So let me copy this. I'm going to copy in this table here of whoa, that looks terrible. Okay, try again. Okay, it might be kind of small if you're looking at it on your iPhone or something like that. But so I'll try and read the important stuff. So, okay, here's a table of several amino acid substitutions in the alpha and beta chains of human hemoglobin. Okay, so let's take a look here. Okay, so here's a hemoglobin chain, maybe from a person from Toronto, one from Oxford, one from Mexico, Bethesda, Sydney, and Saskatoon, which sounds awesome. I'm guessing that might be Canada or something like that. I want to go there just because of the name. Okay, so we have all these different so I guess either the protein was sequenced, probably the DNA was sequenced from individuals from all these different places, and okay, they all have slightly different hemoglobin subunits, mutations in either the alpha or beta subunit, or polymorphisms. We'll get into the difference between why we call something a mutation or when we call it a mutation and when we call it a polymorphism. And so let's just take a look at the hemoglobin from Toronto. So hemoglobin from this person from Toronto has, well, the normal amino acid, okay, is alanine. And now the substituted amino acid was aspartic acid. And I guess that's at position 5 in the alpha subunit. So the 5th amino acid is normally alanine. And in this person, there's been a mutation, or a polymorphism, and it's now aspartic acid. So now what the problem wants us to do is take a look at the genetic code here. Okay, let me copy this in for us. 1 second, I'll pause this. Okay. Let's see, do I have it here? No, I don't have it. Hold on a second. How come I can't get this? I just want to paste this in here. Okay, perfect. Shrink it up here. Okay. Is it recording? Yes, okay, I'm recording. So, now what we want to do is using the code in figure 13.7. I just found this on the internet. This isn't the one from figure 13.7. Determine how many of them can occur as a result of a single nucleotide change. Okay, well, this is pretty straightforward. Okay, so we have alanine. Let's see how many codons are there for alanine. Okay, here's alanine right down here. Is there another? Is alanine anywhere else? I don't see any other alanines. So there are 4 possible codons for alanine: GCU, GCC, GCA, and GCG. Okay, so those are all the possible codons for alanine. Now, for some reason, this person has aspartic acid at this location in the protein. So let's see what their possible codons are for aspartic acid. We have GAU right here in GAC, GAC. Are there any others anywhere? Okay, no. Okay, so let's see. You know, sometimes, you know, people we're not all genetically identical, right? We all have differences in our genomes and our genes, and let's see. let's take a look at the changes that could have occurred between, you know, the gene encoding the alpha subunit in this person relative to wild type to change this one amino acid. So let's see, we have GA, and what we're looking for just one change. Could one change have occurred to change this amino acid from alanine to aspartic acid? And we can see right here, right? So if this codon in most individuals for this alanine is GCC, well, if we had a mutation that changed that C to an A, okay, but 1 point mutation, one little change, this would be a transversion, right? So we're going from a pyrimidine to a purine. So one little change, that C to an A, all of a sudden, now this gene in this person for the alpha subunit codes for aspartic acid instead of alanine. Okay, so just one little mutation and we have changed the protein. Same thing for this one right here, right? If this was the original codon for alanine in the wild type version of the alpha subunit gene, we can change that to A also, and then one single change and it codes for aspartic acid. Okay, so that was pretty simple. I wonder if we really need to go through all of these. I'll go through one more because they're pretty straightforward, and I think it's just getting used to working with the genetic code and seeing that, you know, single mutations like this can change an amino acid in a protein. So let's take a look at Oxford here. So the hemoglobin from the person HBJ, we're calling this for some reason, from Oxford, you know, has a glycine normally, and in this person the alpha subunit encodes an aspartic acid at position 15. Okay, so aspartic acid at position 15 in the alpha subunit instead of a glycine. Glycine is normally found there. In the wild type alpha subunit gene, we have a glycine coded at position 15, and in this person from Oxford, the alpha subunit encodes a aspartic acid at 15. So we'll take our genetic code. How did I do that last time? Let's see. Shrink it up again. Okay, now we need to find all the codons for glycine. So let's see, glycine. Okay, I see a bunch down here, right? So GGU, GGC, GGA, and GGG. Now, any other glycines anywhere? No, I don't see any. Okay, so aspartic acid, again, it looks like there are only two. So GAU and GAC. So any single base changes that can change the 15th amino acid in the alpha subunit from glycine to aspartic acid. So can we find one little mutation? Let's see. Oh, okay, here we go, another one. So same thing as last time, right? We just changed that G. In this case, I think before it was a C, but now we're changing this G to an A. Okay, G to an A. And this right here, our last mutation we looked at was called a transversion mutation, going from a pyrimidine to a purine, or a purine to a pyrimidine. In this case, this is a transition mutation, going from a purine to a different purine. And we can see a transition mutation can still change the amino acid. Both transition and transversion mutations can change the amino acid in a protein. It doesn't need to, but in these two cases it did. Okay, so I think those are pretty straightforward. I guess maybe if I went through some others, we could see some other types of mutations. But I think it's pretty self-explanatory, so I won't do any more of these. And we should also thank, I think it's there's an office here at ISU who is providing the closed captioning for us. So the longer these videos, the more work we make for them. So I won't do any more of the, let me see, there are 1, 2, 3, 4 more examples, and I think they're pretty straightforward. So if you have any questions on those, you can come and see me.